Okay. Kind of a hard day to just do a regular biology lecture. For any of you who were friends with Austin, I'm really sorry, and if there's anything I can do to help anyone, it's not my training, but I'm happy to listen. So everyone remembers that this is exam week, and so there's a review session tonight. All review sessions from now on, I've made an executive decision, have been moved from Morton because Morton sucks. And we're going to be in the teaching lab from now on for review sessions. So tonight from 5:30 to 6:30 is a review session. So we spent a fair amount of time last week talking about plasmids. And remember that what we're really talking about, the big picture here, is genetic transfer and how microbes transfer DNA. And so just to show you why it's so important that we understand how DNA is transferred among bacteria, I want to show you some real-world examples from both the biomedical world as well as the environmental science world, and how it's important to understand genetic transfer in bacteria. So we'll start off with a story that starts before any of you were born. In 1968, there was a terrible epidemic in Guatemala of bacterial dysentery. This is a bacterial infection of the intestinal tract that doesn't simply lead to diarrhea, but this is bloody diarrhea. So this is, I mean, if you think diarrhea is bad, imagine seeing a lot of blood in it as well. It's a very scary disease, and it can be very fatal. None have been more fatal than this particular one was. This particular infection in 1968 was resistant to many of the antibiotics that would typically be used by clinicians to treat this infection. It was resistant to chloramphenicol, tetracycline, streptomycin, and sulfonamides. Never been seen before. 12,000 people out of the 100,000 infected died, which is just a hellacious mortality rate for bacterial disease. Many viral diseases tend to be more virulent, oftentimes more fatal. Even viral diseases don't often have this type of a mortality rate. And part of that mortality rate was due to the fact that it was difficult to treat these infected, many of the dead were children, difficult to treat because of the antibiotic resistance. But the story actually starts earlier than that, back in 1955 in post-World War II Japan. There was a very brilliant infectious disease physician there who noticed when he was treating a woman in Japan that she had Shigella with that same resistance pattern. This actually kind of goes to show why physicians need to be trained as scientists, because this physician was also trained as a bacteriologist and a scientist. He had a PhD as well as an MD. So Dr. Akiba noticed that the woman, when he isolated E. coli, also had the exact same very unusual resistance pattern. So the bacterium that was causing bacillary dysentery, Shigella dysenteriae, a distinct bacteria from just commensal E. coli that lives in the intestine, typically don't have that kind of an array of antibiotic resistances. So he was kind of curious, so he did experiments. So what he did was he went back into his freezer and he got out earlier strains of Shigella dysenteriae, the bacterium that's the etiologic or causative agent of bacillary dysentery. These were isolates that were sensitive to all of those drugs. And when he co-cultured those bacteria with the E. coli that had all four of those antibiotic resistant drugs, what he found was that the Shigella then did acquire the resistance to all four drugs. And they never acquired the resistance one antibiotic at a time. The Shigella always had all four antibiotic resistances or none. There was no partial transfer. It was an all or nothing thing. It turns out that now we're beginning to see, this is one of the first evidences of antibiotic resistance being transferred from bacterium species to a different bacterial species by way of plasmid. So the plasmid in this particular case was called R100, R for resistance. This was in that table of plasmids that I showed you last week. And it has a lot of different antibiotic resistances. Now, the reason why we're talking about plasmids now, and now moving on to conjugation, because this is the way this plasmid is spread, is there's a misconception among many people about how plasmids spread in nature. Many of you in laboratory classes have done transformations where you've been given a plasmid and E. coli cells, and you've put them together and let the E. coli cell be transformed by the plasmid. That is, in fact, the definition of transformation, taking up naked or free DNA from the environment. However, the reason that this gives a misconception is that people believe that this is the way that plasmids typically are transferred in nature, and it's not true. Plasmids are almost never in nature transferred by transformation as you do it in the laboratory. It's a convenient tool that we use in the laboratory, but in nature, it almost never happens. Plasmids are designed, have evolved to spread by conjugation. So this plasmid, the R100 plasmid, looks kind of like this. It has its array of genes kind of packed up into 3 different trunks, or 3 different packages, which literally are operons. It has an operon of genes similar to the F plasmid that we've talked about a little bit and we'll come back to in a few minutes, that dictate the transfer of this plasmid to other cells. It also has all of the genes necessary to replicate itself. And unfortunately for those patients in Guatemala, it also has several different antibiotic resistance genes that it has coalesced together into one region of the gene. And this very simple GIF just kind of shows how this occurs. So here you have, well, of course, I think this GIF didn't start off at the beginning. So here's the beginning. So here's your antibiotic-resistant bacterium with the resistance plasmid, which can now dictate the generation of the sex pilus. So the sex pilus structure and synthesis is entirely encoded by this plasmid. It now can pass on via conjugation a single-strand copy of that plasmid, in this case to a completely different species. So more often than not, this transfer of plasmid is within species. But many plasmids are promiscuous. They don't really care what genus or species or class or phyla of bacteria they find their way into. They just go, and if they can replicate, then great. They've increased their fitness by being able to replicate in a whole different species of bacterium. So now maybe that plasmid is more fit because now it can reproduce in new hosts. So remember that plasmids are entirely selfish elements. They are genetic parasites. And so this is exactly what happened, that a plasmid somehow got passed from E. coli to Shigella, or Shigella to E. coli, or from some bacteria as yet to be identified to start that terrible outbreak in Guatemala in 1968. Now, just a little bit of an aside, because I love the topic of antibiotic resistance, that plasmids are important to understand in terms of antibiotic resistance because of all of the pathways known to give resistance to antibiotics. Plasmids are pretty much the chief carriers, not the exclusive carrier, but the chief carrier of genes that give antibiotic resistance. So if you look at most of the pathways in which a bacterium can become resistant, such as the generation of an efflux pump, many of the genes for these antibiotic efflux pumps, which pump antibiotics out faster than they can come in, or at least as fast as they come in, are encoded on plasmids. So efflux pumps are a great way for bacteria to get rid of tetracycline. Most tetracycline-resistant bacteria have a plasmid, which encodes an efflux pump, which has evolved or mutated over time to recognize tetracycline, and it just gets it out of the cell before it can stop protein synthesis. Other plasmids produce things like beta-lactamases, the enzymes that we talked about that are exported to break down antibiotics. So that's another major way of antibiotic resistance, is the production of an enzyme that breaks down the antibiotics. You can also have things that, mutations that alter the ribosome. So sometimes there are genes in plasmids that encode a different subunit of the ribosome, which now makes that ribosome resistant to antibiotics. So virtually all of the pathways that we know of that can make a bacterium resistant are encoded at some point by a plasmid. So this is part of the reason why these things have spread so rapidly in the past 50 or 60 years, because we've used so many antibiotics. We've put natural selection on the as a premium on the acquisition of plasmids that have these antibiotic resistance genes. But these types of transfers are not simply important to understand if you're interested in biomedical science. These are also very important for environmental scientists, for people interested in bioremediation. So microbes have been found from sites where soils are particularly polluted, where you can find bacteria that can use those pollutants as carbon sources. So one really good example is a bacterium called alkaligenes, or genus called alkaligenes, that was isolated from a landfill that had been polluted. And that strain of alkaligenes had a plasmid within it, which encoded some enzymes that would break down chlorobenzoate, so a pollutant. So the alkaligenes, so somebody thought, okay, we can use this for bioremediation. So they put it in a laboratory simulated polluted ecosystem. The bacterium didn't survive. So alkaligenes just itself didn't do very well with that pollutant in that particular environment. But the plasmid didn't die. The plasmid got transferred to some other bacterium that it could also replicate on, completely different species, and then broke down the chlorobenzoate in that ecosystem. So it's a matter of the plasmid doesn't care whether the host lives or dies. As a matter of fact, conjugation actually helps it find a host that can live in any particular environment. So that previous host dies, well, that's too bad. We've moved on to another host. This actually was used in a real-world example. There was an oil shale underground fire in Estonia years ago, and in the course of putting that out, there was a tremendous amount of polluted water that was generated. So a lot of phenol in that, 100,000 cubic meters of phenol-polluted water. So what they did as a way to try and bioremediate that polluted area was they introduced a bacterium called Pseudomonas putida, which is known to have, or it's most famous for having a plasmid that can drive conjugation, but it has a plasmid that in the laboratory allows that bacterium to break down phenol and polychlorinated compounds. So people thought, great, we can just dump this bacterium in and it will take care of the pollution. Well, it kind of worked. They introduced the bacteria and then found that the phenol concentration went from 25 millikilograms per liter to 9 millikilograms per liter in 6 years, but they could never find the bacterium again. But guess what? They found the plasmid. The plasmid, which hadn't been found in that particular environment before, had been transferred to some bacterium that was much better fit for living in that polluted water underground. And so that plasmid did just fine. It actually helped that bacterium, whose name I don't know, but it helped that bacterium use that phenol polluted water as an energy source. So these plasmids, you may think of them in terms of antibiotic resistance as evil entities, but they can actually do our bidding as well in the case of bioremediation. So these are just a couple of examples of why I think it's important that you understand bacterial genetic transfer. And so, of course, we always have to use the classic pictures of bacteria undergoing conjugation. So here's the sex pilus that's going to drive conjugation, that's going to drive the spread of the plasmid. Now, I showed a picture similar to this before, and what I failed to point out was that this pilus, as impressive as it looks, is not nearly this wide. The only reason that you can see this this pilus in this electron micrograph is that it is literally studded with viruses. There are bacterial viruses. There's an RNA virus that has evolved which will infect E. coli cells only while they're undergoing conjugation. So in effect, think of it as a sexually transmitted disease. If these two bacteria, if this bacteria didn't create this sex pilus to try and spread an F plasmid into this F minus cell over here, these cells would be just fine. But now this RNA virus has bound to that pilus as in injecting its RNA, and so now probably both of these cells are going to be killed by the virus. And lest you think that E. coli can only produce one pilus at a time, so being very impressive, they can produce multiple sex pili at the same time, so they can actually undergo conjugation with multiple cells at the same time. So you can write your own jokes for that. And here's actually a picture of one bacterium utilizing two different sex pili to undergo conjugation with the same cell at the same time. Now, many of these plasmids that drive conjugation, or I should say, all of the plasmids that drive conjugation, have the ability to form such sex pili. So these plasmids are referred to as conjugative plasmids because they can drive conjugation. Now, don't think that all plasmids are capable of conjugation. It's not true. Most of the plasmids that you may ever deal with in laboratory settings, research or otherwise, will probably not be conjugative plasmids. So the plasmids that we use from a biotechnological point of view typically can't drive conjugation. But most of the plasmids that we're interested in that mediate genetic transfer do so by conjugation. Those other plasmids, by the way, actually hitchhike. They can actually utilize conjugative plasmids. If they're in the same cell, they can kind of hitchhike a ride into another cell when that cell goes through conjugation. So they're kind of like parasites upon parasites upon parasites. But we're going to talk, for the most part, about the paradigmatic example of a conjugative plasmid called the F factor, or the fertility factor. That's the plasmid that's driving the synthesis of those very impressive sex pili that I'm very fond of showing pictures of. These plasmids, when they exist inside of E. coli cells, actually have two different modes of replication. So they have a mode of replication, as I mentioned earlier, called the theta replication, and a rolling circle replication. When that E. coli cell containing an F plasmid is in an environment where there is no F- cell present, it's just all itself, all by itself, F plasmid cells as far as the eye can see. In that particular case, the bacterium is not going to go through conjugation because it has no potential partners. In that particular case, as those E. coli cells divide, the F plasmid replicates by theta type replication. And it does so by starting replication at this site here called the ORI-V. V stands for vegetative. This is the origin of replication that the plasmids use during vegetative growth of the E. coli cell. So this is where the replication forks will begin, and they'll proceed to the right and to the left, and eventually you get a structure that looks like the Greek letter theta, and then eventually you get two circles. Now, the situation changes completely when an F-positive E. coli encounters or detects in the environment an F- E. coli cell. So these plasmids are so highly evolved they can actually detect an otherwise genetically identical bacterium that lacks the plasmid. There is a receptor on the surface of normal E. coli cells that the sex pilus binds to, that the bacterium can actually detect. What happens is that when a bacterium takes up this F plasmid, one of the things that this F plasmid does is it represses the expression of that receptor. That outer membrane protein that's used by bacteria to detect F- bacteria is no longer expressed, or expressed at much lower levels. This way, the bacteria with the F plasmid don't waste time trying to accomplish conjugation with cells in their environment that already have the F plasmid. That wouldn't be beneficial. Why waste your resources trying to conjugate with somebody who already has the plasmid? So the way that the bacteria detect uninfected, if you will, uninfected by this F plasmid, is the presence of that receptor that's not yet repressed. So that's actually the receptor, turns out to be the receptor for the pilus as well. But when that cell is detected and conjugation is going to be initiated, a whole series of gene expression changes occur. But one of the things that occurs is that the mode of replication of the F plasmid changes from vegetative to rolling circle. So in rolling circle replication now begins at this ORI-T site. T stands for transfer. That's the way to remember it, because at that origin of replication, rolling circle replication is going to occur, and that's going to result in the transfer of a single-stranded copy of that plasmid being transferred across the conjugal pore into the F- cell. And you'll notice in this cartoon, it's a little bit subtle, but look at the ORI-T, you see that it's an arrow. Because in rolling circle replication, Once once replication begins at this site, it goes in one direction only. From the vegetative origin of replication, replication forks go in both directions. In rolling circle replication, what's going to happen is that an enzyme called the relaxase enzyme, or TRA-I, T-R-A, capital I, you'll see it in a slide in a minute, will actually nick, that is cut, one strand at the of a phosphodiester bond at that ORET site, and then the rolling circle replication is off and running, in this case going upward, peeling off a single-stranded copy which can be transferred into the other cell. So rolling circle replication is used during conjugal transfer between cells. It's not used if there's not a target cell in the environment. If you look at the entire F plasmid, the region that we're really interested in right now, we've talked about other regions. So what's not shown on here would be the CCDB, CCDA, that addiction module that we talked about. That's encoded somewhere else in this chromosome. But the region that we're talking about now, which will drive everything related to conjugation, is found right here in a single operon. And these are all of the genes in the operon. So let's one by one go through these genes. No, I'm kidding. I can't do that. I don't know what most of these genes do. But I mentioned that there is an enzyme that is triggered, is induced when the bacterium, the F-positive bacterium, finds an F-minus bacterium. That's this TRA-I gene shown here on the right, encoding an enzyme relaxase. That gene doesn't normally get turned on until there's a cell in the environment that's an F-minus cell. Then this operon gets turned on, including TRA-I, which will nick at ORET to initiate translocation. The other proteins that are in here, such as TRA-K, B, and P up here, these are the proteins that make up the components of a secretory pathway that we'll talk about, because this is a very complex process that the bacteria and the plasmid has evolved to transfer a copy of itself. And then the TRA-A is the pillin protein. So this is the subunit protein that's polymerized together to make that long sex pilus. And remember, it's contractile. So when you see pictures like I like to show you of that really long pilus, don't think for a moment that there is DNA being transferred through that long pilus. It's not. That pilus will now be depolymerized from the base, literally pulling that F- cell closer and closer until they actually come into contact with one another. Then a pore will form between those two cells. And other proteins that are encoded in this TRA region or transfer region will drive that translocation. So all of the genes that are necessary for conjugation fall within that 30 gene operon, shown here as the TRA operon. The F plasmid, and this is true for many plasmids, but it's very well studied in the F plasmid, has some genetic parasites upon itself. So remember, one of the big take-home pictures that I want you to be able to remember when you run into me in 10 years at homecoming is that you have this still constant idea that plasmids are not a normal part of the biology of bacteria, they're parasites. But now, that parasite actually has on it parasites as well. These things shown here as ISC3, or ISC3. I spent too much time in that building this weekend. Insertion sequence 3, IS3, there are 2 copies of this genetic parasite. This is a mobile piece of DNA. It's like a transposon, but even simpler than a transposon. It does nothing. It encodes, we'll talk about these in more detail in another lecture, but insertion sequences basically encode nothing but a transposase enzyme that allows them to move from place to place to place. They don't do anything else. They don't have antibiotic resistance. They might, if they randomly land in the middle of an important gene, they might mess that gene up. But in and of themselves, they're pretty harmless, but they're parasites. They're selfish organisms, entities. But there are 2 copies of those on there, and here's a completely different type of insertion sequencing called IS2, insertion sequence 2. Because and I'll show you this in a subsequent slide, these parasitic pieces of DNA also exist, not just on the plasmids, but on the bacterial chromosome themselves. So there are identical copies of IS2 and IS3 on the chromosome of E. coli. And what that does is that sets up a situation where you have nearly, if not completely, a perfect match between a stretch of several hundred base pairs of DNA on the chromosome because of the two ISC3s. That can drive homologous recombination. Remember, REC-A, my favorite enzyme, has the ability to bind to DNA and to find homologous DNA and to accomplish generalized recombination or crossing over. What this means is that F plasmids, or basically any plasmid, which has sequences that are shared between it and the chromosome, has the ability to be what's known as an episome. An episome is a genetic character that can exist either free in the cytoplasm of the bacterial cell or be incorporated into the chromosome of the cell. And they can move back and forth between those two lifestyles, either incorporated into the genome or living free as a circular form. So that's what this cartoon is trying to show. So here are, although they're shown in different colors, just to differentiate the fact that this is a plasmid-borne copy of the IS element. And here is a chromosomal copy, which is, if not identical, pretty damn close to identical. And because they're so close to identical, REC-A can drive homologous recombination, make crossovers, break that DNA, reseal the two DNAs together, and now you can see that this F plasmid can be incorporated in its entirety into the chromosome of the cell. It doesn't hurt the plasmid at all. Nothing is lost. It's just in a different location. Every one of the genes necessary to drive conjugation still exists. There is still, although they're not shown on here, there's still an origin, well, I guess this is what O stands for here, the origin of replication, the ORI-T. This O would actually be ORI-T for transfer. So now what could happen is in this particular arrangement shown at the bottom, if this bacterial cell comes into contact with an F- cell in the environment, it still behaves exactly the same way. It tries to conjugate with that cell. It's going to induce that relaxase enzyme. So TRA-I is going to nick at this origin of replication. And you can see from the arrow that rolling circle replication is going to drive replication to the right. But now, instead of being a small circle as an F plasmid, now it's part of a big circle. The chromosome, the bacterial chromosome is circular as well. So now that rolling circle replication gets out of the plasmid portion of the DNA and into the chromosome. This cell is what's referred to as a high frequency of recombination cell. Because now when conjugation occurs, it's not just the F plasmid that's going between cells, but part of the bacterial chromosome. So this is the first time where we're actually talking about bacterial sex, because there is actually now going to be exchange, potentially exchange anyway, of bacterial chromosomal DNA. Not just the parasitic DNA, not just the plasmid DNA, but the actual chromosome of the DNA. And you notice the arrows in this go in both directions. So almost as easily, if not as easily as this plasmid was subsumed into this chromosome, the reaction can go the opposite way because you still have two IS elements in fairly close proximity, maybe 50 or 60,000 base pairs apart, but that's nothing. Well, it is a lot, but this can drive recombination, and so the plasmid can resurrect itself out of the chromosome back exactly as it was before. So this probably happens at some frequency. So the plasmids go in, they come out, they go in, they come out. Because to the plasmid, it really doesn't, in a way, it doesn't really matter. Now, that conjugation that's going to transfer these things is a very complex array of proteins put together. So all of the proteins here, not just the sex pilus, but all of the ATP binding, the energy generating molecules that create, that polymerize this sex pilus and depolymerize it and assemble the whole thing. These are all proteins that are encoded in that TRA operon. So this becomes actually, you can think of this, although we already talked about protein secretion, this is actually a secretion mechanism. But in this particular case, what's going to be secreted is going to be DNA. We talked about protein secretion. But for this type, this is referred to as a type 4 secretion system. This is actually the conjugation system. So what's going to be passed through here is going to be DNA, the F plasmid. So this is the process that the F plasmid drives for conjugation, and it's probably quite similar to conjugation in other bacteria. So on the top, you have an F positive cell and an F minus cell that it detects in the environment. Because remember, the F minus cell has a receptor that the F positive cell doesn't have. It's been repressed because of the presence of that F plasmid. The sex pilus contacts and binds to that receptor on the F minus cell, and then it depolymerizes from the base of the F positive bacteria, literally pulling the F minus bacteria over to it, where it can now not use that sex pilus at all, but instead form this pore, which is shown here in the blow-up. And you can see the complexity of this. So H, I, and J make up much of the machinery. But here's the relaxase, the TRA-I gene, which is going to nick this DNA. So at this point, this double-stranded plasmid is now bound by this type 4 secretion system, TRA-H, I, and J. It's cut at one strand only. That's the definition of a nick. It cuts one phosphodiester bond, and now what can be passed through there during rolling circle replication is that single strand that's going to be passed into the other cell. That's what's being shown here in 4. This one strand goes in because this other strand is being peeled off by rolling circle replication. So as DNA polymerase is copying this light blue strand, it's peeling off one strand which is going through. Once that copy has gone through in its entirety, both single strands are recopied. And so the end result are 2 F positive cells. So the only thing that's been transferred here, though, is parasitic DNA. It's just the F plasmid. So this is not, truly a sex act, because from the bacterium, from the host's point of view, there is no advantage to this. Nobody has increased their genetic diversity. Nobody has potentially acquired some kind of an advantageous mutation, unless this particular plasmid has an antibiotic resistance gene on it, for instance, which many times they do. And so if that antibiotic is in the environment, then there's selection to allow that. So it's not just antibiotic resistance either. Oftentimes there are operons for things like catabolic pathways or breakdown pathways as well. So this is, in a nutshell, how typical bacterial conjugation takes place. Now, as kind of an aside, because what I'm interested in is how bacteria cause disease, some bacterial pathogens have actually co-opted that conjugation system. So as we just described it, that system was designed to pass plasmid DNA, parasitic DNA, into another cell. But many bacterial pathogens have actually made evolutionary constructs of this pathway. And so they use a type 4 secretion system not to pass DNA from cell to cell, but instead to pass toxic molecules from the bacterial cell into a host cell. So this is an example from my favorite bacterium in the world, Helicobacter pylori, the stomach pathogen that causes cancer. This particular bacterium has taken what used to be a conjugation system to pass plasmids and evolved it, changed it, mutated it, so that now instead of producing a sex pilus to pass DNA, instead what it does is it still creates a pilus that's a hollow pore. This hollow pore, which can be, again, just like the sex pilus, polymerized from the bottom up and literally grows out, will interact with an alpha-5 beta-1 integrin on your host, on your stomach cell walls, and will form a pore, will form a direct conduit, a hollow pore between the cytoplasm of the bacterium Helicobacter pylori and the cytoplasm of your stomach cells. And then what gets passed across is a toxic molecule called CAG-A. CAG-A is a really fascinating, huge protein that gets into a eukaryotic cell like a stomach cell, and it just goes it makes the cell go absolutely haywire because the cell recognizes it as one of its own proteins. It phosphorylates it. It turns on all manner of signal transduction pathways, changes the morphology of the cells, changes their secretion patterns. Actually, is believed to be responsible for the cancer that can be caused. This CAG-A protein is actually referred to as an oncoprotein. It has, by means that are still being investigated, the ability to promote tumors in the human stomach. But this used to be evolutionarily just a plain old system for passing plasmids back and forth. But somebody got a hold of it and just changed this, just repurposed it to now be used as a pathogenic mechanism and a really wicked pathogenic mechanism. But anyway, back to conjugation. So again, conjugation almost always, driven by plasmids. There are some transposons that have all of the genes necessary to drive conjugation, but I think they stole those from a plasmid. So what's shown here is on the top is an F positive cell and an F minus cell. The sex pilus is formed, rolling circle replication, passes over a single-stranded copy. It's recopied, the second strand in the recipient cell, and so now you have an F positive, you have two F positive cells. So they contact the F minus cell. That contact is what induces the TRA operon to make the sex pilus, initiates rolling circle replication. And that F minus cell is referred to as the transconjugate. So that is the recipient molecule. And so in an F positive, F minus conjugation, the resulting transconjugate will always be F positive. These are great exam questions because you're going to see it's not quite as simple always as the conjugation between an F positive and an F minus cell. There are other examples. So that single strand F DNA is resynthesized in the transconjugate to make it an F positive cell. And that F factor will now repress the sex pilus receptor in that F minus cell, so nobody else will bother to conjugate with that cell in there. So very efficient. Now, these plasmids are what are referred to as self-mobilizable. You can see why. They have all of the machinery necessary to drive their own spread from cell to cell. But other plasmids have actually evolved to kind of take advantage of these conjugated plasmids. Rather than encode all of the genetic material to drive their own conjugation, they just kind of wait for a conjugative plasmid to get into the cell with them, to cohabit that cell. And this turns out to be a fairly common situation in the environment. Most environmental bacteria have at least one form of a plasmid. Many have 2, 3, or 4 different plasmids all at the same time. So if only one of those plasmids is capable of driving conjugation, there's at least a possibility that during that conjugation, driven by that conjugative plasmid, that some other plasmid, simply by mimicking that ORI-T sequence, can get itself transferred. Because if a non-mobilizable plasmid, one that doesn't have all of the TRA operon to it, if it simply has that short DNA sequence that TRA-I, the relaxase enzyme, recognizes, it's going to nick that and then transfer one strand across into the other cell. So these cells are these plasmids are very highly evolved parasites. They actually just kind of hitchhike along the way. Okay, come back to this episome situation. So remember that the F plasmid, like many plasmids, can exist within the chromosome of the cell. And I mentioned that this cell is now referred to as an HFR, or high frequency of recombination cell, when it has this type of an arrangement. The reason why it's called a high frequency of recombination is because now when mating occurs, when conjugation occurs, what can be transferred is not simply the F plasmid. In fact, for the most part, the F plasmid in its entirety never gets transferred in this mating. But you can actually have bacterial DNA exchange. So this becomes a really important mechanism for bacterial evolution. So the acquisition of new DNA from the environment is oftentimes done by this HFR F minus type of a conjugation. Because remember that the HFR cell has the entire F plasmid, it's just now subsumed within the larger circle of the bacterial chromosome. So it can still detect an F minus cell, It can still synthesize all of the proteins necessary to make the sex pilus to undergo rolling circle replication. It's just that now when it nicks that DNA at ORET, instead of nicking in a small circle, it's nicking a big circle. It's nicking in the chromosome. And so now you see here in red, the first portion of the DNA that's being peeled off is part of the F plasmid. That's what the red is representing, the F plasmid. And that can get passed through the conjugal pore into the other cell. But as this goes around the circle, it now runs quite quickly into bacterial chromosomal DNA, and it transfers that just fine. Now, in nature, so in the laboratory, under perfect conditions, you can do an experiment where you can take an HFR E. coli cell and an F- E. coli cell, and if you don't even breathe, if you just make things very, very still, you can actually get the entire transfer. You can get rolling circle replication to go all the way around the 4 million base pair chromosome of E. coli and transfer the entire DNA in it. It takes about 100 minutes. Matter of fact, if you look at old genetic maps of E. coli, you'll notice that genes were mapped in minutes. That was the way that people used to determine where genes were. Gene X tended to come over in a mating at 10 minutes, whereas gene Y didn't come over until 40 minutes. But in nature, because this conjugal pore is relatively delicate, any kind of a shear force, any kind of movement tends to break it quite easily. So more often than not, when you have a transfer of DNA from an HFR cell to an F- cell, that F- cell remains F-. Because for it to become F positive, the rolling circle replication would have to go all the way around the chromosome to pick up the last portion of that F plasmid and transfer it. The chances of that happening are virtually nil in the real world. So that's why the transconjugate in an HFR to F- mating is always going to remain, virtually always going to remain F-. That doesn't mean that it's going to be unchanged, because this DNA, the bacterial DNA that came over in there, probably has some degree of match with the chromosome. So by homologous recombination, it can switch into the chromosome. It can replace alleles on that chromosome with perhaps beneficial mutations, more often than not probably deleterious mutations. So this HFR to F- is very important in terms of generating bacterial genetic diversity. Because that, and again, I got ahead of my bullets. The contact of an HFR cell with an F- cell still does all of the same things in terms of inducing that operon and the transfer. But almost never will the entire F region get sent over to the transconjugate. So the transconjugate almost always remains F negative. So let's see if I can make this work, because this is somebody else narrating conjugation besides me.
Plasmids that mediate mating are found in several bacteria. One example is the F plasmid in E. coli. The F plasmid contains genes directing synthesis of a pilus that contacts the recipient F- cell, lacking the plasmid. The plasmid can be transferred from an F+ cell to an F- cell. A nick is noted in the DNA to start transfer, and a single strand of the plasmid DNA is transferred into the recipient. DNA replication in both donor and recipient replaces a missing strand. If the F plasmid integrates into the chromosome by crossing over, an HFR cell is formed. Now, the chromosomal DNA tags along with the F DNA when it is transferred into the recipient cell. After the single-stranded DNA enters the recipient cell and replicates, it can be combined with the resident chromosome. In this HFR cross, chromosomal genes can be transferred from one E. coli cell to another during mating. In an HFR cell, the F plasmid can excise from the chromosome to reform an F+ cell. Sometimes the excision is not exact, and the chromosomal DNA becomes part of the F plasmid. The resulting plasmid is called an F prime plasmid. After transfer of an F prime plasmid, such as the F-like plasmid, the recipient cell has a duplication of the chromosomal DNA of the F prime plasmid. The duplication of DNA makes F prime plasmids useful for complementation analysis.
So you can see how complicated this gets, because we haven't even gotten to the F primes yet. So we've talked about a mating between an F positive and an F minus cell. We've talked about the fact that that F plus, or that F plasmid can go into the chromosome, generating an HFR cell, which can still mate with an F minus cell. But now, as that animation began to show, is that sometimes weird stuff happens. And so when that plasmid comes back out of the chromosome, so more often than not, this HFR cell is going to resurrect itself as a perfect F plasmid, just like what went in, because it's going to undergo recombination using this IS element and this IS element, because they're closest to one another. But occasionally, if there's another IS element further downstream, you can get recombination between this IS element on the far right and one that's not being shown in here, say on the other side of this locus called A. That will still generate a circular molecule resurrecting itself from the chromosome. It will still have all of the F genes present, so it will still be truly an F plasmid, still capable of driving conjugation, but it's going to carry with it part of the bacterial chromosome. So that's what's going to be called this F prime plasmid. So in this particular case, what they're showing is that the locus A has been taken up into this F prime plasmid, which has now come over to an F minus cell and created it to be an F prime cell. But now this cell is almost certainly diploid, a situation known as merodiploidy. So it has two copies of a gene that is otherwise haploid for. Remember, bacteria are haploid organisms. They have only a single copy of genes. In this particular situation, as it's outlined here, this bacterium may actually have two copies of that A locus. So it would be what's referred to as merodiploid. So mating between an F prime cell and an F minus cell can still occur. It will still occur. And what gets transferred is that F prime factor. An HFR cell can never conjugate with an F prime cell because this plasmid still has all of the F genes. It's still capable of repressing the expression of that receptor molecule. So this cell is not recognized as a target for conjugation. So a mating between an HFR and an F prime cell can never happen. Also a good exam question, kind of a trick question. I don't like trick questions, but sometimes I feel a little mean, and so I'll throw them in. So think about that. Now, so here is a conjugation, exactly as we're talking about, between an F prime cell and an F minus cell. Now, this F prime factor, this F prime plasmid, which has part of the chromosome, the locus of chromosome, it doesn't leave part of the F plasmid behind. This is an error in this particular cartoon. But now it can drive conjugation with an F minus cell, and what gets transferred over is an F prime. And so now you have a cell which has two loci of A. And so if one is advantageous over the other one, the bacteria may reap some benefit from having this increased genetic diversity and increased copy in that particular allele. So that F prime cell, because it has all of the F plasmid cells with it, or F plasmid genes with it, can still drive conjugation. It just now has some extra material with it, so that it makes the new cell, the transconjugate cell, merodiploid. And that new cell would be referred to as F prime cell. Not F positive, but F prime. So this conjugation can actually be promiscuous. So we're talking about conjugation here with the F plasmid going from E. coli to E. coli. But at some frequency, because there are so many bacteria and there's so many different other bacteria in the environment, at some real frequency, this occurs where the F plasmid or some other plasmid can now be transferred to a completely different species. So this will make for very rapid evolution in a bacterial population or in an ecosystem, because now you have these plasmids that gain benefit by actually being transferred to a completely new species. Okay, and then when we come back, next time, we'll stop talking about conjugation, I promise, and we'll move on to talk about transformation.